he attenuated this virus in the lab to generate a virus that didn't cause disease, but that was highly immunogenic. So when you immunize people with this attenuated virus, you induce amazing immune responses, but it wasn't causing disease. And so fast forward 70 years, to this day, that is the vaccine that is licensed as the yellow fever vaccine. That's in fact licensed by Sanofi. Max Tyler got the Nobel Prize for this work. And in fact, this vaccine is now considered to be one of the most successful vaccines ever made. 600 million people worldwide have been immunized. If you immunize a person, you induce immune responses that last for more than 3 decades. And it induces amazingly high titers of neutralizing antibodies and cytotoxic T cells. So this was a gold standard of what could be achieved in vaccinology. And so we thought, like 12 years ago, why not use this example, this gold standard vaccine, to deconstruct the immunological mechanisms by which it was working? Why not reverse engineer this vaccine and then use the principles to design new vaccines against HIV, malaria, and many, many other pathogens for which we don't have vaccines? So one of our earliest observations was to show that, in fact, this virus, this attenuated virus, was activating the innate immune system through multiple toll-like receptors expressed on dendritic cells. And these multiple toll-like receptors were important for mediating the immune response to this vaccine. So that was an important insight that we had. And so that then led to other work in which we collaborated with bioengineers to design synthetic nanoparticle-based vaccines that effectively did the same thing that this yellow fever virus was doing. It contained many different TLR ligands. But at the same time, around that time, we said, well, here's a vaccine that works. It's been licensed. It's been given to 600 million people. Well, why don't we do an experiment in humans to look at the immune responses induced by this vaccine in great detail? And at that time, about 12 years ago, systems biology was coming into fashion, but it hadn't been applied to vaccines. So we thought this would be a cool experiment to do. So this is what we did, a clinical trial, trial number 1, in which we vaccinated healthy adult people with this yellow fever vaccine. Blood samples were collected at all these different time points, some very early after vaccination, day 3, day 7, day 15, day 60, day 90. In Rafi Ahmed's lab, they measured the tetramer positive CD8 T cells induced by this vaccine. We also measured the neutralizing antibody induced by this vaccine. And what we did was to analyze in great detail the innate response induced by this vaccine. And what we saw was that there was a very strong transcriptional signature. And working together with Alan Aderam and Eva Lee and colleagues, what we could define was a transcriptional signature that was very typical of a viral infection, a lot of interferon genes and antiviral genes. And in a sense, that was expected. But keeping in mind that this experiment had never been done before, it was kind of reassuring to see that indeed this live viral infection was causing an antiviral signature. But more importantly, we were able to define a signature of gene expression that could discriminate between high responders and low responders in terms of the subsequent adaptive response. Also, working with Eva Lee, we used machine learning techniques to define a focused set of genes that could then be used to predict the magnitude of the T cell response and the antibody response in a completely independent trial, trial number 2, that was done 2 years after the completion of the first trial. So this provided proof of concept evidence that in fact systems biology approaches can be used to study immune responses to vaccines, and they can be used to define signatures that predict the immunogenicity of this vaccine. Now, for those of us not in the vaccine field, the significance of this is that as you develop vaccines, and the perfect example is the COVID-19 vaccines that are being developed, some 135 plus vaccines that are in the pipeline. The big question is, how do you go about testing all these vaccines? Well, the standard practice is to do preclinical studies, and then a phase 1, and then a phase 2, and then a very, very large and expensive phase 3 trial. And typically this whole process can take up to 15 years and more than $500 million. So you can imagine, although this has been extremely successful in the past, in the future, as we develop more and more vaccines, clearly this kind of protracted and expensive approach cannot be tenable. So the idea that you could have a system where you could define early biomarkers of vaccine efficacy was really an important challenge in vaccinology. And this study, I think, provided the first proof of concept that that was possible to do that. Subsequently, we and a number of different labs, in fact, extended this kind of approach to analyzing immune responses to other vaccines. You've heard a beautiful talk from John Sang in which he mentioned some work his group has been doing on the flu vaccine. We also looked at the flu vaccine. And then in a few minutes, you're going to hear from Shuzhao Li, who's now at Jackson. At that time, when he was, we were privileged to have Shuzhao as a postdoctoral fellow in our lab several years ago, in which he did this amazing work in which we had been looking only at the transcriptional profiling, but what Shuzhao did was to devise a way in which he could integrate multi-omics data sets, not only from transcriptomics, but also from metabolomics, plasma cytokines, and high-dimensional flow cytometry to construct a multi-scale, multifactorial response network in which you could use to understand immune responses to vaccine, in this case, shingles vaccine, and that was Shuzhao's paper that was published back in 2017. So the other implication of this, this approach is that not only can you predict the strength of the immune response to vaccine, but you can also predict how effective a vaccine is. And that may or may not have something to do with the strength of the immune response. Obviously, if you have a higher antibody titer, you'd like to think that that should be a better vaccine, right? But not necessarily so, because there are many different types of immune responses that we may not be measuring in our standard assays that contribute to the effectiveness of a vaccine. Now, one approach vaccinologists have used to assess efficacy is shown here. So this is an example of a study with that we did with GlaxoSmithKline, a vaccine company, to test their malaria vaccine. It's called the RTSS vaccine, in which healthy people were given this malaria vaccine once, twice, and then 3 times. And these healthy people were then deliberately challenged with mosquitoes infected with Plasmodium falciparum. That is the malaria parasite. And you can do this in this way. You see, this is a study participant, and you can see in this Chinese takeaway jaw, he's been exposed to mosquitoes that have been infected with Plasmodium falciparum, and then you can deliberately infect them. That's Bill Gates, by the way, standing just a few feet away. I was amazed that they allowed Bill to be in this room at the same time. But in any case, so this challenge model, this vaccine gives you about a 50% efficacy. So 50% of the people get infected, 50% are protected. What we were able to do was to collect blood samples at all these different time points, do an in-depth analysis of the transcriptional profile, and to define a signature that correlated with protection. And so this illustrates the notion that not only can you have signatures that predict immunogenicity, but are also capable of predicting efficacy. Okay, now this kind of challenge model is also available for other viruses such as influenza, norovirus, bacteria such as typhoid, and now, as you might have heard, there's even discussion of establishing this controlled human challenge model for COVID-19. So I want to spend a few minutes turning to the biological insights that we could gain from this kind of massive profiling of the immune responses to vaccines. And I just want to highlight one example. And this came about from a study that we had done with the seasonal influenza vaccine that was published back in 2011, in which we saw that a gene encoding toll-like receptor 5, which, as many of you know, senses bacterial flagellin. These are these flagellin molecules in bacteria. This is a receptor, toll-like receptor 5, expressed on dendritic cells that senses this bacterial flagellin. We saw that, in fact, the expression of that gene was contained in a signature that correlated strongly with the antibody response to the seasonal flu vaccine 30 days after vaccination. You see, this expression of this gene peaked at around 3 days after vaccination. There was a very strong correlation with the antibody response 30 days after vaccination. So that was published back in 2011. So this raised the obvious hypothesis: why should there be a gene encoding TLR5 induced by a vaccine, a viral vaccine? Flu is a virus. Could this have some biological significance? So Jason O in our lab followed up on this. He went back to the mouse model in which he used wild-type mice or mice genetically deficient in toll-like receptor 5, and he immunized these mice, and he showed, wow, there was a marked diminution in the antibody response to the seasonal flu vaccine. Then Jason said, could it be that this TLR5 was sensing bacterial flagellin from the gut bacteria? And so he treated these mice with antibiotics, and then he saw, again, there was a marked diminution in the antibody response. He also did the experiment with germ-free mice that are devoid of any bacteria from birth, and he saw that there was an impaired antibody response. So this provided evidence that, yes, toll-like receptor 5 was critical in inducing antibody responses to flu, and that this was because they were sensing flagellin from gut bacteria. Of course, this was done in mice. That was published in 2014, but this raised a larger question in vaccinology. That is the microbiota important in immune responses to vaccines? Now, why is that an important question? Well, because first of all, if you look at the composition of the gut microbiota in populations that live in different parts of the world, in fact, there's now quite a bit of evidence that populations have different compositions of gut bacteria. So this is one study published in PNAS in 2010, in which they looked at a population in Burkina Faso versus the European Union. And you can see, even at a high level, they're very different compositions of gut bacteria. And yet, all these different populations get the same vaccine. It's not as if we have a different vaccine that's given to this population versus this population. So the obvious question is, well, what is the impact of this difference in microbiota on vaccine efficacy? And that's not the only study. As I mentioned, there's quite a bit of evidence now. There have been other studies that have shown that the gut microbiome varies by age and geography. So to address this, what we did was a human clinical trial that we set up at Emory. This was set up back in 2015. And really, this is the brainchild of Nadine Rupail, who's an outstanding infectious disease clinician at Emory University. And the postdocs in my lab who led the analysis are shown here: Thomas Hagen and Mario Cortez, two outstanding young postdocs. Mario has now moved on from the lab. And so the study was a very simple study. It was a small study in which there were 11 control subjects. So these are healthy young adults between the age of 18 and 45, and 11 antibiotics treated people. So these are people who are not infected with any bacteria, but they're healthy people who have been given antibiotics. So they're given a cocktail of neomycin, vancomycin, metronidazole. You can imagine this is a pretty strong regimen of antibiotics, and you can imagine Nadine had to do a lot of talking to convince the regulatory board that we need to do the study. Neomycin targets gram-negative bacteria, vancomycin targets gram-positive bacteria, metronidazole targets anaerobic bacteria. These healthy young people were administered these antibiotics for 5 days. And then one day before the last day of antibiotics, they were immunized with the seasonal flu vaccine. The immune responses in these people were followed from baseline 3 weeks prior to antibiotics administration to the day of the administration, the first day of administration, all the way to 1 year. And we collected blood samples, and we did an extensive analysis of the innate and the adaptive responses using a number of different techniques that are shown here. We also collected stool samples and analyzed in great detail the composition of the gut bacteria. And so what did we find? This was published just, I think, about 4 months ago. And I'm just going to summarize the findings in a simple cartoon. What we found is shown here. This was a perspective piece that we had to write on this. But essentially, the message could be summarized as follows. The gut bacterium does have an effect on the innate and the adaptive response to vaccine. However, this effect was only apparent in people who had had very little previous exposure to influenza vaccine or to an influenza infection. Let me clarify what I mean by this. You see, the first study that we did in 2015, which I described to you 2 slides ago, we didn't bother to check whether these participants had had the flu vaccine the previous year or the year preceding that, or for the few years preceding that. Neither did we ask these people, did you have anything that resembled flu infection in the previous year? And as we analyzed the data, we realized that the majority of these people had very high levels of baseline titers. By baseline titers, I mean the antibody titers against influenza, which exists even before you give the influenza vaccine. This antibody titer is presumably because of pre-exposure to influenza vaccine from the previous season or influenza infection at some point in the past. Now, we found in the 2015 study, the effect of antibiotics seemed to have very little effect on the innate or the adaptive response to this vaccine. So in the following flu season, in 2016, we did a second study in which we deliberately excluded people who had received the flu vaccine for the 3 preceding years prior to this, or had been infected with flu for 3 years prior to this. And there we saw a very substantial effect of the antibiotics. In fact, there was a very substantial drop in the antibody response and major changes in the innate response. So this told us something very important about the human immune system. That is, the human immune system is adaptive. That's why we call it the adaptive immune system. It can withstand even the most severe dysbiosis. What do I mean by dysbiosis? It's a disruption of the gut microbiome. It truly is adaptive when it has pre-exposure, immune memory. And so this raises the obvious question, what is the impact of the microbiome on immune responses to a primary vaccine? Suppose we did this study in which humans had never seen this vaccine before or had never been exposed to the pathogen before. What would it be? What would the results be? And also, as a corollary to this question, what about neonates in the first few weeks of life or infants in the first year of life? I mean, the immune system of these special populations are pristine. They are nascent, they're evolving, the repertoires are evolving, and the microbiome is nascent and evolving in the first year of life. And yet, these infants and neonates are being immunized with no fewer than 15 childhood vaccines. What is the impact of the microbiome on the responses childhood vaccine? This is a study that we're now doing in the Gambia in Africa and in South India, 2 places where, unfortunately, antibiotics usage is so prevalent that by the time a child is a year old, there's a high likelihood that this child has been exposed to antibiotics. What is the impact of this on the vaccine response? No one knows, and we're beginning to address this issue in these 2 trials. So I want to conclude this segment of the presentation by summarizing, and this is very much ongoing work, and this was a review that we published back in 2010, in which we thought, at that time, there was a lot of discussion between discovery-based basic science and applied science, as there still is today. And in many ways, they're very, very different enterprises. But we felt that it was important to view them as a sort of continuum, as an iterative exercise, where, the linear translation from preclinical animal models such as mice, the non-human primates to humans was fine, but it could also go the other way. You could, in fact, start your experiment with the human population, as we did with vaccines, use the tools of systems biology to understand signatures of vaccine immunity and efficacy, work together with computational people, and then generate interesting new biological hypotheses, such as the one that I talked to you about, and then test these hypotheses in animal models, and then obtain better insights about the mechanisms by which this vaccine was working, and then those insights would ultimately help you design better vaccine. So the example I've given you today is really this, starting with the flu vaccine published in 2011, in which we saw that correlation with TLR5, getting into the mouse mechanistic studies in 2014, that now raised a whole set of questions about whether this could happen in humans. So this was then tested in humans. Now, I didn't have time to dwell on the mechanisms, but we've discovered that in fact certain metabolites are critically involved in orchestrating the antibiotics-mediated effect and how they affect immune responses to vaccine. And in particular, bile metabolism seems to be a critical player. So this has now raised a new mechanistic project in the lab where we're working in knockout mice that are deficient in bile sensing receptors and so on. Hopefully sometime in the next 2 or 3 years, we could do a sensible human trial in which you supplement humans with bile, secondary bile, and see how this affects the immune response. So really, this sort of, I think, framework is important in general for vaccinology, but also, I would say, biology in general, that it's really, the applied science and discovery science are kind of two sides of the same coin. Now, in the remaining 15 minutes, I do want to talk to you a little bit about some ongoing work in using systems approaches to understand the immune responses to COVID-19. Now, here we are in the middle of this pandemic, and I checked yesterday, I think there are some 13 million infections in the planet. And the thing to remember is that, of course, viruses have been emerging as far back as history can remember. I mean, you can go back to Athenian times and you can see things like the Antonine plague or the plague of Justinian that pretty much killed half of Europe, 30 to 50 million people back then. And then, the famous bubonic plague, that killed like 2/3 of the population of Europe. And then more recently, just 100 years ago, the Spanish flu that killed about 50 million people. And so here we are today with COVID-19. And just to remember that this won't be the last pathogen to emerge, I think, for thousands of years until we have some sort of a magic bullet that prevents pathogen emerging, that we will continue to have these pathogens. So around March or so, we started looking at immune responses in COVID-infected patients. We were very fortunate to be able to get some samples from Professor Malik Piris in Hong Kong University and also from Emory University, in which we analyzed the immune responses in COVID patients who had been in the ICU, or patients with severe disease, or mild or moderate patients. And we used a number of different techniques. We used CITE-se. CITE-se is mass cytometry that, it's high dimensional mass cytometry, RNA-seq, bulk RNA-seq, O-link, bulk RNA-seq, but also single-cell RNA-seq, O-link, metabolomics, and then the antibody response. And the questions were, what is the immune response to COVID like? Is there an immune signature that can predict disease severity? What is the immune response in asymptomatic individuals? So first, let me show you some data from CITE-se. These were 40 samples from Atlanta, 100 samples from Hong Kong. And what we see in this UMAP is that, let me point some some interesting features. First of all, in COVID-infected patients, we see a massive expansion of the plasmablast response. These are the B cells that make lots of antibodies, and massive activated CD8 T cells. That was from the Atlanta cohort. Very similar results in the Hong Kong cohort, so that's good. Whether you live in Hong Kong or in Atlanta, the immune profiles seem quite similar. We also saw reduced frequencies of plasmacytoid dendritic cells, both in Atlanta and in Hong Kong. These are the dendritic cells that fight viral infections, that make interferons. And also what we saw, this didn't work, but anyway, so within plasmacytoid DCs, what we saw was this molecule called mTOR. mTOR is mammalian target of rapamycin that is a central metabolic sensor in the mammalian immune system. It's involved in many, many different physiological processes, and mTOR was severely impaired. mTOR activation was impaired in this plasmacytoid dendritic cells. Why this was so interesting was back in 2008, we had shown that in plasmacytoid DCs, this mTOR was so important in mediating interferon alpha. Interferon alpha is the cytokine that fights viral infection. So in COVID-infected patients, we asked the question, well, so is it true if these cells, blood cells, are these plasmacytoid DCs, do they have less capacity to produce interferon alpha? So we did this experiment. We got PBMCs from COVID-infected patients. We did a very simple experiment, and we just stimulated them in tissue culture with a toll-like receptor 7/8 ligand. And then, so this is without any stimulation here. This is a healthy person. Now, this is after stimulation of PBMCs from a healthy person. Here, you can see very high levels of interferon alpha, lots of cells making interferon alpha, also some TNF as well. That's from a healthy person. Now, this is from an infected person. You see, this is without any stimulation. With stimulation, substantial reduction in interferon alpha. And by the way, this FACS plot here is I'm gating on the plasmacytoid dendritic cell, okay? So you see, you can see lots of patients here, whether you are in the ICU or severe or moderate or healthy. You see, there's this marked diminution in interferon alpha. Then we said, okay, you know, everyone says COVID is an inflammatory disease and you're measuring all these cytokines, IL-6 and all these things in the plasma. But what about the cells in the blood? Are those the cells that are making these cytokines? So we did the same thing. We got these PBMCs from healthy people or from infected people. And you know, without stimulation, you're looking at TNF alpha on this axis, IL-6 on this axis, nothing. You don't see any TNF or IL-6 from the PBMCs from infected people. Now, what about after you stimulate with TLR ligands? Okay, you see, wow, lots of cells make IL-6 and TNF alpha if you stimulate a healthy person, cells from a healthy person with TLR ligands. But look what happens if you stimulate cells from an infected person. Not much. It's pretty dramatic. And so this is with bacterial TLR ligands. This is with viral TLR ligands. Again, you see quite a bit of cytokines in a healthy person, but not much in an infected person. So again, the myeloid cells, myeloid DCs in COVID-infected patient seem to be compromised in their capacity to produce pro-inflammatory cytokines. And why is that so? Well, we looked at one of the key mediators, transcription factors that mediate cytokines, pro-inflammatory cytokines, NF-kappa B, and you see reduced activation of NF-kappa B in those myeloid cells. So the cells in the blood cannot make much interferons, or they cannot make much pro-inflammatory cytokines. However, we looked in the plasma to see what about those, can we detect those cytokines? And we saw, whoa, as reported by many other publications now, many of them in bioarchive, there's lots of IL-6 in the plasma. You see more in the ICU patients, severe patients also have some, moderate, not much, healthy, not much, more TNF-alpha, more MCP3. But we also saw 3 new molecules that hadn't been reported that correlated beautifully with disease severity. NRAGE, which binds to the RAGE receptor, which is expressed on lung alveolar type 1 cells, TNF-SF14, also called LIGHT, which binds to lymphotoxin beta receptor, and oncostatin M. Again, these 3 molecules are strongly correlated with disease severity. Why this is so exciting is because NRAGE and LIGHT have been shown to be involved in lung inflammation in the context of other inflammatory diseases. So this raises the prospect that this might be good as therapeutic targets with COVID. Now, to analyze this further, what we did with CITE-seq analysis, John introduced you to CITE-seq, so I'm not going to tell you what this is. We analyzed 8 COVID subjects, 3 severe, actually 9, 3 severe plus 1 ICU, 3 moderate, 2 convalescent, 5 healthy subjects, and 2 subjects infected with influenza or respiratory syncytial virus, which are 2 other respiratory pathogens. The way we did this CITE-seq was to first enrich for dendritic cells, which form a very, very minor population. So we enriched for these artificially and then added them back to the PBMCs so that now they are represented at a higher frequency than they would have been otherwise, okay? So then we did the CITE-seq analysis, and you can see in this UMAP clustering, you can see we could define these cellular clusters very nicely, you know, T cells, platelets, B cells, myeloid cells, and as you might expect. And the first thing to see is that the COVID-infected patients could be distinguished very clearly from healthy patients, okay? Or from flu in RSV patients. And the second thing was that when we looked at the different disease states, severe, moderate, and healthy, again, we could see gene expression profiles in certain clusters that could discriminate between these different disease states. I want to highlight one biologically interesting aspect that emerged from this. You know, I mentioned to you that when we analyzed the capacity of blood cells to make type 1 interferons, that COVID-infected patients were compromised, that plasmacytoid DCs didn't make type 1 interferons in COVID-infected patients. So we said, well, if that is true, let's look at the interferon alpha an interferon beta message in these UMAP clusters. And you can see we saw 0, no interferon alpha, no interferon beta. But the surprise came when we saw what we call ISGs, the interferon response genes, or IRGs. For example, IFI27 and IFITM3 were expressed at high levels in certain of these clusters, including monocytes, some monocyte subsets, and some myeloid DC subsets. And so this was a surprise. How come that they are expressing these interferon response genes, even though there's no interferon alpha, which is needed to induce these? So we dived into this more deeply, and let me lead you through these two heat maps. This is looking at the CDC2 cluster, which is a subset of dendritic cell. This is the plasmacytoid DC cluster. This is looking at COVID patient 5, 6, and 7 in purple. Those are patients who have moderate disease. And you can see those 3 patients have high expression of all these ISGs, IFI27, DDX60, and so on. These are all ISGs, okay? However, if you look at the severe patients shown here in red, COVID-2, COVID-1, COVID-3, those have markedly less expression of these ISGs, okay? And convalescent patients shown in green don't have very much, and healthy people do not have these. Now, the same kind of phenomenon is seen with plasmacytoid DCs. So this suggested that, well, maybe these ISGs are expressed in moderate patients. But to confirm this, because after all, we only had a few subjects, we did independent analysis of bulk RNA-seq, of bulk PBMCs in 18 subjects, okay? And we looked at these ISG gene expression signatures. And again, what we saw was there was a correlation with not only disease severity, a new factor that emerged was time post-onset of symptoms. So yes, disease severity was important in the sense that moderate patients did tend to have greater ISGs, but even more important was the time post-onset of symptoms shown here. So this beautifully confirms our CITE-seq analysis data. This is bulk RNA-seq with bulk PBMCs, okay? And you can see this time effect here. So you see there's an early brisk, actually this is to confirm this at another level. We said, let's analyze in the serum of interferon alpha using the SIMOA technology. SIMOA is this new technology which is based on an ELISA method, highly sensitive at measuring very low concentrations, femtogram per mil amounts of interferon alpha. And you can see that there's this initial burst of interferon alpha and then it goes back down, you see? And if you look at the SIMOA, the data, that is the amount of the interferon alpha in the plasma on this x-axis versus the ISG score in the CITE-seq on the y-axis, see this beautiful correlation that beautifully confirms our CITE-seq analysis, okay? Finally, what we saw in our CITE-seq analysis was there was reduced HLA-DI expression in myeloid DCs, especially in the severe patients. We went back to the CITOF data and say, is that really true? And indeed, yes, strikingly diminished expression of HLA-DR in both the Atlanta patients and in the Hong Kong patients in myeloid DCs, but also in monocytes, both myeloid DCs and monocytes, okay? And lastly, we said, I mentioned to you this molecule called NRAGE that we detected in the plasma. I mentioned to you that binds to the RAGE receptor on lung alveolar cells. And we said, well, what about the expression of NRAGE in these CITE-seq analysis? We see this cluster of monocytes express high levels of NRAGE. And in fact, the higher the lower the expression of HLA-DR, the higher the expression of NRAGE. So we think we've defined a subset of monocytes found in the blood that is very low in expression of HLA-DR that's expressing this NRAGE molecule. And that correlates with what we see in the plasma. So the last piece is, how is all of this happening and why are we seeing lots of inflammatory cytokines in the plasma, but we see this refractoriness or this impaired capacity to produce pro-inflammatory cytokines by these blood cells? We said, well, the lung is not a sterile environment. The lung, especially the upper respiratory tract, we have a lot of bacteria. Could it be, how do these bacteria play into this pathogenesis of COVID? So from the plasma of these COVID patients, we analyzed the endotoxin levels, and we saw this remarkable finding that in a certain proportion of severe and ICU patients, there was high levels of endotoxin in the plasma compared to moderate or healthy patients. We also isolated bacterial DNA from the plasma, and then we measured the amount of bacterial, we use 16S ribosomal RNA to measure bacterial DNA. And again, you see high levels of bacterial DNA in plasma from these severe infected people. So how is that in any way impacting any of the immunological measurements we've seen? What we see is that the greater the concentration of bacterial DNA on this x-axis, or we can do this exercise for endotoxin as well, the higher the amounts of pro-inflammatory cytokines, interleukin-6, TNF, MCP3, TNF superfamily 14, oncostatin N, and NRAGE. So this would suggest that in understanding the pathogenesis of COVID, it's not just the virus that seems to be having this effect. We also have to factor in bacterial DNA. Now, how exactly are these microbiota influencing the cytokine storm? What kind of bacteria is this? We're analyzing that now. So this really summarizes what I've said here. I think what this data suggests is that it's instructive to view the pathogenesis of COVID as occurring in two different stages. First, early on in infection, you have this mild disease, and there's this initial burst of type 1 interferons, not from the blood. We cannot see this from the blood. We think this is coming from the lung, but then that circulates. Type 1 interferons can circulate everywhere, and this switches on a transient burst of ISG interferon response genes in blood cells that we saw in our CITE-se. We also saw in the bulk RNA-seq. As the disease progresses, you now get excessive inflammation, lots of TNF, IL-6, and many, many other pro-inflammatory cytokines, again, coming from the lung, not from the blood. We don't see this coming from the blood. There's inflammatory cytokines, also bacterial products. This, we feel, gives rise to this suppressed innate immunity that is in the blood. That is, type 1 interferons in PDCs is low, pro-inflammatory cytokines are low, and what we think are these myeloid-derived suppressor cells, those HLA-DR low cells that seem to be quite refractory to innate stimulation. So I think this highlights an interesting concept about the immune system. You know, I think about this. So there's a rampant infection in the lung. That's an organ where there's massive inflammation. Yet, light years away, in a galaxy far, far, far away, things are very different. There seems to be suppression of the innate system. So how is that so? We call this spatially dichotomous immunity. Why would a biological system work in this way? We can imagine perhaps that if there's an infection in the lung, it's better to confine the inflammation to that site and really to prevent the rest of the immune system elsewhere from becoming excessively activated, because that could be deleterious to the host. Now, the bigger question is what we've seen with COVID, does this apply to other kinds of tissue infections, other kinds of respiratory diseases, severe respiratory diseases like MERS or SARS-CoV-1 or H5N1 influenza? We're actually in the process of investigating those questions now. So let me thank the people who did the work. Lots of people collaborated here. The COVID work was led by Prabhu Arunachalam, Florian Wimmers, and this was done in collaboration with Malik Piris's lab and Nadine Rupail's lab. And the early work was done by many others, including Shazali. So thank you.
All right, thanks a lot. Thank you very much, Bali. This is an amazing amount of work that you were able to do, I think, within this past few months. All kinds of different data modalities in the COVID. I have some questions about that part, and but in the chat box there are a few questions. Maybe first we can. Yeah. So the first one is, I think you've given an example on it, but on the practical side, are there examples of vaccine formulations that were changed after insights from systems vaccinology?
Yes, it's. From the practical side, I think we're working very closely with vaccine manufacturers, including GSK, to use the systems biology data to guide adjuvant design, vaccine design. Something that I didn't have time to talk about today was the idea that adjuvants such as AS03 have a long-lasting imprint on the innate immune system, epigenetic changes. That has implications in terms of how we think about adjuvants. So adjuvants are not merely promoting stronger and longer lasting adaptive immune responses, but they may themselves be having long lasting effects on the innate immune system. So that is something that I think many vaccine manufacturers are interested in, and they are thinking about how to now begin to translate these insights in terms of practical outcomes. So I think the field is still in a state where a lot of insights have emerged, but the translation into the clinic is still happening, and we're not yet there, I would say.
Yeah, I mean, there's, for anything, I think there's a delay between discovery and application, but I think it's promising for several vaccines at least. And I mean, we are learning a lot more for vaccinology with the COVID trials. I think it's going to help. So another question about, I had a similar question, but what do you think about, I mean, now you are linking, I think the TLR5 work was interesting, the flagellants, but then it also helps in the viral infection. So how about the other way around, which is BCG and COVID, I think, where you have, what do you think about the BCG vaccine being potentially helpful for COVID?
Yeah, no, I think it's very interesting, and I'm very keen to see the outcome of the clinical trials that are ongoing. Certainly the idea that, I mean, that the whole study is based on the premise that immunization with the BCG vaccine will imprint a heightened state of antiviral response on the, by activating the innate immune system. And this is coming largely from the work of Mihai Netia and colleagues. And I think the data so far has been very, very intriguing. As I mentioned to you, we're seeing some similar effects with adjuvants, but let's see. I mean, the ultimate proof is in a clinical trial. So I think let's all wait and see how this pans out.
So maybe a few COVID questions I will take. Given that we are into the break, feel free to leave, guys, if you want to leave. But it's while we have Bali, I think it's a great time to ask a few more questions. So for COVID, do you speculate that bacteria strain distribution in patients may be related to their disease severity? I think you hinted at that, but
yeah, that's a great question. So, you know, the data I showed you shows that yes, in the plasma, you do find high levels of endotoxin and bacterial DNA in COVID-infected patients with severe COVID disease. We are working with a company to do metagenomic sequencing to analyze the composition of the bacteria to see where these bacterial products are coming from. Are they coming from the lung, or are they coming from the intestine? We don't have any data yet, but that's an important issue that we're looking into.
I guess I'll be selfish and ask my questions because it did. So for your site of work, and when you are describing these different cell types in the COVID case, so have you looked at the T cells and tried to see whether they have some exhaustion kind of marks to whatever the exhaustion means in the COVID patients, severe or convalescent?
Yeah, no, that's again a very good question. We, you know, traditionally we've always tended to focus on the innate immune system, and so we've sort of you know, the lab feels they're already so busy looking at monocytes and DCs and so on, and you know, so we only, I mean, we've looked very sort of superficially at T cells, and we see signs of effector T cells, strong activation of effector T cells. So we haven't dived into the T cell side of things, and a lot of other great labs are doing that. So we figured that we should focus on the innate system. So.
Yeah, no, I mean, we are also working on the T cell side of things.
Yeah.
So it would be great to talk to you about that.
Yeah.
There are questions about COVID affecting different organs, right? We generally think about respiratory, but other organs such as reproductive system, and so would considering any of these other targets be useful for vaccine design? Is I think one question.
Yeah, well, you know, I think the entry point of the SARS-CoV-2 virus seems to be through the respiratory tract. But it is true that there are emerging reports of COVID infecting cells in other organs. In fact, I saw a report in bioRxiv that it may even affect neuronal cells, infect neuronal cells. And, there have been other reports of infection in other tissues and so on. So I think it's still very early days. We don't know the extent to which this infection becomes more systemic and the circumstances under which it becomes systemic and the impact of that on pathogenesis and immune responses. So, when I said that this is a tissue-specific infection, of course, that's an oversimplification. Yes, it starts off in the lung, but it has far-reaching effects on other sites outside of this tissue, either as a result of the virus itself getting out into other sites, perhaps, and the evidence for that is, I think, still emerging. I mean, I think people have done PCR assays, but to my knowledge, no one has yet isolated virus that can replicate from other tissues, and indirect effects through microbiota, metabolomics, and so on. So I think, it needs to be looked at from a much broader systems context. It's exactly a system. This kind of infection is exactly what systems folks like you guys should be looking at. It's systems really at the level of all the different sites in the body.
Thanks. So maybe one or two more questions quickly. So I think this depends on the sample size, and everyone wants to look at it. But female-male differences and then different ethnic groups, right? We know that they are different. They are different risk for COVID.
That's a great question. So the analysis of CITE-se and the analysis of the bulk RNA-seq was done from the Atlanta cohort, which predominantly, the vast majority of people there were of African-American descent. And so that's important to know. And indeed, there's emerging evidence, as you may know, that there may be differences in how different ethnicities respond to this infection in terms of disease severity. So certainly that's something that would be important to address in the future. So ethnicity, age, and other variables as far as the populations are concerned. I think it would be very important to extend this sort of thing into much larger studies that can address this.
Yeah, I think we all need larger sample sizes. I mean, 100 patients that you have is that's an amazing number at this point. But to study other factors, I think we'll need to go into the several hundreds or thousand. Maybe one last question that's not COVID, and then I will leave you to answer them in the chat box, or we have the Cafe Connect feature. If you can join, that would be great. So the cancer vaccines and microbiome connection. So what do you think we should be looking at? That is the question, I think.
Yeah. I mean, I think there's been some very elegant work on that in the last couple of years. There's a couple of very important papers published in Science about 2 or 3 years ago, showing that actually 3 papers back to back, showing that in humans, the microbiome did play an important role in how people respond to checkpoint blockade therapy, and that it could be influencing immunity to cancer. So those were pivotal studies. And I think based on some of the work that I showed you with the other kinds of vaccines, there's no question that the microbiome is going to be playing an important role in modulating immunity to cancer vaccines. And that's something that really needs to be looked at in more depth.
Certainly, there are many groups doing that. Thanks a lot, Bali. This has been a great talk.